Thank you for the introduction, and thank you for the opportunity to present my work here. Today I'll be talking about the causal contributions of beta and gamma oscillations in motor control. Effective motor control depends on a delicate balance between facilitation and inhibition. And many neurological and psychiatric disorders, this balance is disturbed. For example, in Tourette syndrome, there is too little inhibition, resulting in involuntary motor tics. Or like in Parkinson's disease, where there is too much inhibition, which can result in a slowness of movement that is so extreme that patients even feel like they are glued to the floor. So we know that a cortical basal ganglia network is involved in maintaining the balance between inhibition and facilitation. However, it is still unclear how the nodes within this network actually communicate with each other. Recent evidence points into a direction for beta and gamma oscillations, but having an opposing role. The beta oscillations have been associated with slowness of movement and inhibition, whereas gamma oscillations seem to promote voluntary movement. In this study, we set out to determine if these beta and gamma oscillations are actually causally related to motor control and are not just merely epiphenomenal. To do this, we recruited a sample of 35 participants between the age of 20 and 30 years old and applied transcranial alternating current stimulation, or TACS, in a 20 or 70 hertz stimulation frequency. So with TACS, a current flows between the electrodes in a particular frequency. And this has been shown to entrain the endogenous brain oscillations in that stimulation frequency in the brain. And thus gives you experimental control about the strength of these brain oscillations. So in this study, we placed the electrodes over the pre-supplementary motor area in a 1x4 montage, which has the advantage that it's the stimulation area is more focalized, and it also doesn't induce phosphenes. Concurrently, we recorded a 128-channel EEG, and we have 5 minutes of resting EEG before we started stimulating. We have the EEG during the stimulation, and also 3 minutes after the stimulation session. I won't be talking about the EEG during the stimulation today because we're still in the process of refining the artifact removal procedure. But I'm happy to discuss all of this after the talk or maybe tonight during the poster reception. So participants were performing a stop signal task during the stimulation. And this type of task taps into both inhibition and facilitation and works as follows. Participants see an indicator moving up and are instructed to press a force button whenever this indicator reaches the 2 yellow target lines. However, sometimes they get a stop cue before this indicator reaches these target lines, indicating that they should not press the button anymore. We evaluated the effect of the stimulation on both force measures, so the force that was used to press the button, and response times. Important to know is that the stimulation was in an intermittent fashion, meaning that 2.5 seconds before this indicator started filling up, the stimulation was turned on and remained on for 5 seconds. After that, 1 or 2 trials followed where no stimulation was given. And this was repeated throughout the experiment. So when we have a look at the results on the raw force data, we can see that the total force that was given on the force sensor was indeed lower during the 20 hertz session than during the 70 hertz stimulation session. However, we did not find any significant differences between when the stimulation was on or off. This suggests that there might have been some lasting effects of the stimulation in the non-stimulated periods, or that there is just large variability between the participants. So first, let's have a look at these potential after effects. When we sorted our behavioral trials or non-stimulated trials into trials that were following the stimulation directly, or those trials where there was first a non-stimulated trial and then the non-stimulated trial again, then we see that there is a significant effect between stimulated trials and non-stimulated trials with one non-stimulated trial in between. but not between stimulation and the non-stimulated trials directly following them. And also in the EEG, after effects were evident. So when we compared the beta power in the resting EEG before we started stimulating and after the stimulation session, we saw a significant increase in this beta power when we stimulated in the beta range, so with the 20 hertz frequency, but not when we stimulated with the 70 hertz frequency. Besides these after effects, we also noticed that there was a large variability in how much force participants used to press the button. And this means that it might not be a grand average, might not be the best way to represent the data when looking for effects between stimulation and non-stimulation. So instead, we reflected the data in a person change way, in the sense that this line here, the dashed line at 0, is the performance on the non-stimulated trials, and then compared how much more or less force there was during stimulation. So all these dots are individual participants, and what we can see is that actually during GO trials there was a slight increase in force during 20 hertz stimulation, and a strong decrease as expected during the successful stop trials. For 70 hertz stimulation, we still did not find a difference between on and off stimulation. So what is also evident from these person change plots is that there is a very high variability in how the participants responded to the stimulation. About 70% went into the direction as expected, but the other 30% did not respond. So one potential factor for this variability might be that we did not individualize our stimulation frequency. So we used our resting state before we started stimulating to define the peak frequency in the beta range for these participants post hoc. What we then observe is that the participants that had a definable peak between 18 and 20 hertz, so 2 hertz less or lower or higher than the stimulation frequency, actually tended to be the ones that responded in the way that we expected. And the ones that fell outside of this range did not, with a few exceptions that are marked in red. So what about the stimulation effect on the response times? Again, here we found a main effect in frequency in the sense that the stop signal reaction time was faster when we stimulated in a 20 hertz frequency than when we stimulated with 70 hertz. But again, we did not find any differences between on and off stimulation. So for this, we decided to use a computational model, which actually includes the response times and accuracies of these trials of all individual trials, and is able to pick up on subtle differences in responding. So in this case, we made use of a drift diffusion model, which was developed by the group of Tim Verstijnen in Pittsburgh. And it works as follows. So the GO process, this green line, is modeled as a single variable which is accumulating towards the decision boundary here on top. When the GO process crosses this decision boundary, a response will be made. However, at the same time, the stop process is modeled as a competing signal. So the stop process is accumulating towards its own decision boundary, and if the stop process reaches this decision boundary before the GO process reaches its own, no response will be made. So this model actually captures the balance between facilitation and inhibition. Context, or in our case, stimulation, can modulate 3 different parameters in this model. So first of all, the onset delay of when this process starts accumulating can be modulated. The decision boundary could be changed, or the speed of the accumulation process could vary with the stimulation. When we fitted these models for both our 20 and 70 hertz stimulation session, we saw that in both cases, this speed of the accumulation process was provided the best fit to our data, although in opposing ways. So for the 20 hertz stimulation session, the rate of accumulation was decreased, so the GO process was slowed down. And for the 70 hertz condition, we saw an increase in accumulation rate, such that the go process was speeded up. So to sum this up in a few take-home messages, our results indeed pointing towards the direction of a causal role for beta and gamma oscillations in motor control that are opposing. We saw that beta stimulation slowed down voluntary movement, whereas gamma stimulation seems to facilitate it. And in my opinion, it should be used more often. These type of computational models could be used more often to look into the underlying processes of our stimulation effects on behavior. I'm still a big believer in the power of an intermittent stimulation design, but it is important to take into account that you might have some lasting effects of your stimulation train. So it might be important to increase the time between the offset of the stimulation and your next baseline non-stimulated trial to not cloud your effects due to after effects in the data. And lastly, it might be worth investing some time in determining the individual frequencies to stimulate at, such that you have a better odds of actually entraining the brain in the way that you would like to. And finally, I would like to thank all my collaborators and funding agencies for making this all possible.
Thanks very much. Thanks very much. We have time for a few questions. Very, very interesting. I was curious about the location of stimulation, which was pre-SMA, and I don't necessarily have the best knowledge of the subtleties of the difference between all the premotor regions, but since the task, it seems, that you used was very visually cued, I would think that stimulation over maybe dorsal premotor or even ventral premotor would be useful. Do you think maybe that has something to do with the variability that you see?
Well, the main reason why we chose pre-SMA is that when you do an fMRI based study with this task, you mainly see the pre-supplemental motor area and in right inferior frontal cortex being involved in the inhibition of the process. And that's also why we went for that in this particular case.
Oh, yeah. That makes sense. Thank you.
Okay, yeah, this is time to close this session, and thank you very much. And we thank you again for 6 excellent presentations and also the participation of the audience. Thank you very much.